Hello everyone, and welcome to today's live broadcast. Fine-tuning of myosin activity shapes actomyosin network organization in tissue contractility of the C. elegans Spermatheca, presented by Alison Wirshing, graduate student at Northeastern University. I am Marjorie Torres of Labroots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by Labroots. Labroots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the button in the bottom left corner and follow the process to obtain your credits. Please join me in welcoming Dr. Alison Wirshing. I will now turn the presentation over to her.
Hello, good morning everyone, and thank you Marjorie for that lovely introduction. So again, my name is Alison Wirshing and I am a graduate student at the Cram Lab at Northeastern University. And what we're interested in the Cram Lab is how do contractile non-muscle cells perceive and appropriately respond to mechanical stimuli. For example, in humans, there are smooth muscle cells that surround the cardiovascular system and myoepithelial cells that surround glands. In the vasculature system, smooth muscle cells must be able to perceive and appropriately respond to changes in the luminal pressure in order to maintain tube diameter. Similarly, in gland cells, myoepithelial cells must be able to perceive when the glands are full and then contract to expel gland contents. One of the ways we know that cells respond to mechanical stimuli is by reorganizing their actin cytoskeleton. So here you can see an example of confluent cells together. Upon the application of shear stress, now there are production of these parallel actomyosin bundles or stress fibers that help the cells withstand the stress. Similarly, if you pull on two attached cells, mechanoperceptive proteins will recruit cytoskeletal components and reinforce those junctions. The actin cytoskeleton is also reorganized under different conditions. Here I'm showing you an example of asthma experiment where they took bronchial epithelial cells and exposed them to dust mite irritation. And now what you see is, especially if you look right there, in the center of cells, they have begun to become very contractile and produce those actomyosin bundles. So how is it that actin and myosin is able to generate forces within cells to withstand external forces? This is accomplished through myosin motor activity on actin filaments. So here in this cartoon, I'm showing you myosin in green and actin in red. And what that looks like at the molecular level is here shown at the bottom. So in non-muscle cells, myosin activity is really tightly regulated by phosphorylation. When the myosin regulatory light chain is not phosphorylated, such as is shown here, it is kept in this inactive conformation. It cannot interact with actin and it cannot produce force. However, upon phosphorylation, it now adopts this active conformation, can form bipolar filaments, which then pull on actin and slide actin and produce forces. So that is how myosin activity on contractility is regulated at the molecular level. However, in addition to the level of phospho or active myosin, we also have actin-myosin network organization really impacting tissue and cell contractility. So now if we zoom out and think of actin and myosin as an entire network, you can appreciate that actin network orientation as well as connectivity can influence contractility. So here, my actin network is not very well oriented, and now I'm going to get sort of a global uniform contraction. However, if I have a very aligned network, I'm going to predominantly get contraction in one direction. If, on the other hand, I think about connectivity, if I have a very not very connected network, I'm going to get small local contractions. You can imagine if I cut the rope that those green men are pulling on. Instead, if I have a very connected network, now I can get global contractions across an entire cell and across entire tissues. And so the system we use to study how cells are able to regulate their amount of active or phosphomyosin and their actin organization in order to appropriately maintain tissue contractility is C. elegans reproductive system. So the C. elegans reproductive system is basically one long contractile tube. It is composed of two U-shaped gonad arms, shown here and here, that come together into a common uterus in the center of the animal. So during the reproductive life of the animal, the oocytes develop in the distal gonad, which is shown here. As they come around the bend, that's shown in that yellow loop, they mature. And then once they are ready, sheath cell contractions, the tissue shown in yellow, push the oocyte into the spermatheca. The spermatheca is stretched, the embryo becomes fertilized, and then is expelled into the uterus. And so here's just a zoomed in picture of what that looks like, where the spermatheca is shown in green, the sheath cells that surround the oocyte and contract to push the oocyte into the spermatheca is shown in yellow, and the uterus is shown in red. So specifically the part of the system that we're really interested in and that we use as our model is the spermatheca, which is a sac-like organ of 24 myoepithelial cells that houses the sperm and is the site of fertilization. We like to use the spermatheca for a few reasons. One, it has discrete and predictable events of being relaxed, of being stretched by the incoming oocyte, and then producing coordinated contractions after fertilization. So all of our stretch and contraction experiments happen in vivo without us having to manipulate the system. Additionally, this has to happen 150 times per spermatheca, meaning the system must be very robust, and all of this is easily visualized in live animals. And so to give you a feel of what that looks like, I'm going to show you a movie of an ovulation. So this oocyte here, you can see is outlined with a dotted line, is going to be pushed by the sheath cells, shown in yellow, into the spermatheca. It will become fertilized, and then spermathecal contractions will push that embryo into the uterus. So we'll watch that now. All right, pushed into the spermatheca, it's becoming fertilized, and now exiting through the spermatheca uterine valve into the uterus. So I've been saying that during this process, the spermatheca becomes dramatically stretched and then has to contract. And so maybe that is easier to see if you look at what the cell outlines actually look like. So in an unoccupied spermatheca that does not contain the oocyte, you can see the cells look very relaxed, sort of compressed. And then once the oocyte enters, the cells become really stretched out. And so what we're interested in is, what does the actin cytoskeleton look like in this process, and how does it contribute to tissue robustness and contractility? So to address this, we first set out to say, okay, what does the actin network look like? On the top here, I'm showing you a cartoon. And I'm showing this because one of the things that's really nice about studying this system is that each cell in each spermatheca develops the same way in each animal. So they always have the same 24 cells, and those cells always have the same neighbors. And in this cartoon up here, which you can see as the arrows in each cell indicate the alignment of the actin network. So it's very stereotyped, it's the same across every animal. And what you can see is that the actin network becomes aligned along the long axis of each cell. Interestingly, these parallel actin fibers are also present even in the unoccupied spermatheca. So although they're probably easier to see here in the occupied spermatheca, that's because the cells have been stretched out. It's not because they're not absent any longer when the spermatheca is no longer occupied. And then if we look here at a cross section, you can see in blue I have false colored the sperm, And then in here in pink, I have false colored the oocyte just to show you what it looks like when it's stretched out. And you can also see, if you look carefully, that these bundles, these actin bundles that I've been talking about, are at the basal cell layer. Okay, so now you know what actin looks like, end of story. Except previous graduate student Ismar made this really interesting observation that in young animals, these actin bundles that are parallel that I've been talking about are absent. So our question was, all right, what is happening? How is it that these actin bundles are forming? Is this something that happens as part of development, such that as the animal matures and prepares to undergo these stretch and contraction events, the actin cytoskeleton develops? Or is it actually that something about ovulation, something about stretching the cells and having them contract is what causes the actin network to go from something that's very webby and isotropic and not organized, shown here, maybe clearest if you look at that one cell, to an actin network which is highly organized and has nice parallel fibers, again, running along the long axis of each cell. So to address this question, we used two different techniques to disrupt oocyte entry in order to uncouple animal age, so is it just maturing, from actually an ovulation event where you have stretch and contraction leading to these fibers. And so to do this, we either genetically blocked oocyte entry by disrupting a gene required for the production of oocyte. The other technique we used was to block sheath cell function. Therefore, there are no sheath contractions to push the oocyte into the spermatheca. And what I hope you can appreciate is that in all cases, compared to the control organized actin cytoskeleton, if we disrupt oocyte entry, so if we block ovulation, we never develop those nice parallel organized fibers. You can see that another way across many animals if we quantify anisotropy, which is just a measure of how oriented the network is. And so here you can see that regardless of animal age, they never develop organized fibers if you block oocyte entry. So to answer the question, no, it's not just something to do with development, but actually ovulation is required to go from a disorganized network to an organized network. So what actually happens during ovulation? A lot of things, and work from our lab and others has really gone into understanding what are the pathways involved in perceiving cell stretch and regulating spermatheca contractility. And here I'm just highlighting some recent work from our lab, and I know this is a lot of information, but really what I want you to take home from this is that all of the key players are highly conserved. So both calcium signaling and Rho signaling are conserved in C. elegans and in mammals and in your favorite contractile cells. So this isn't just some weird thing that happens in worms, it's very highly conserved. But instead of going over all of the details in that glorious pathway, I'm instead just going to show you the simplified one because I don't have time to talk about every member. And here I'm just going to bring up the key players that will be important in this talk. So as I mentioned earlier, we have the calcium side of the pathway and the Rho side of the pathway. So phospholipase C, shown here at the top, is responsible for calcium production, which then, through its interaction with calmodulin, increases myosin activity through activating the myosin light chain kinase. More kinase activity allows for more phosphomyosin and more contraction. On the other side, we have the Rho pathway. So Rho becomes activated by cell stretch, which then activates Rho kinase, which then deactivates the off switch, myosin phosphatase. So without myosin phosphatase, we again have an increase in active phosphomyosin and increased contractility. So in this way, cell stretch through two sides of the pathway, one regulating the on switch, one regulating the off switch, leads to increased contraction. And so one of the ways we've been able to study this is by visualizing calcium in vivo. So here I'm going to show you a video. Pardon me. And in this movie, what you'll see are the cell outlines of the spermatheca in red and calcium in green. We use a genetically encoded calcium biosensor, GCAMP. So an increase in fluorescence intensity indicates an increase in calcium. So now the oocyte is entering the spermatheca. Now the cells have become stretched out, and you can see those big calcium waves going back and forth across the tissue. And if we measure calcium intensity or fluorescence intensity over time, what you can see over this 50-minute time is that it's very stable and doesn't change at all, except during ovulation. So once the cells become stretched out and produce a contraction, that's when you have that peak of calcium. So really it seems that calcium is very tightly regulated and is important for contraction. So what we know up to this point is that the Spermatheca becomes highly stretched during ovulation. We also know that during ovulation we have production of calcium transients that really peak at the onset of contraction. And we know that something about either cell stretch or calcium signaling or something to do with ovulation is required to go from this disorganized isotropic network seen in young animals to this highly organized, very parallel actin network seen in mature adults. So now our questions are, when exactly during ovulation does this occur, and what is the molecular mechanism driving this organization? And so it became clear that in order to address these questions, we needed higher temporal resolution. So we needed to be able to see what's happening in real time. And so to answer this, we turn to in vivo imaging. So what I'm going to show you here is a movie much like the one you saw earlier, except now I'm using a tissue-specific promoter driving GFP-labeled actin. So the only tissue you're going to see is the Spermatheca. And although you can't see it, there is a proximal oocyte that is ready to be fertilized right here. And what's going to happen are sheath cell contractions are then going to push the oocyte into the Spermatheca. The Spermatheca will become stretched out by the oocyte, and then dramatic actin reorganization is going to take place. Okay, so let's see if we can get that to play. And cool. Go. All right, and stretched, and now contraction is happening, and now you can really see those beautiful fibers that I was talking about earlier. We're very clear now. I'm going to play that one more time, and what I want you to try to see this time is the difference now immediately after entry and then at the end of the movie. And that's going to be important on the next slide. Okay, so I briefly mentioned this earlier. We have this tool called Fibril tool, which we are able to use to measure the organization of the actin network in individual cells. So here I'm just showing you what an experiment might look like. So I would take a movie and then shown in yellow would outline an individual cell and then measure the increase in actin organization over time. And so what you can see is that early on, we have very low organization, very isotropic, not organized network, and then later on it becomes organized. And so then down here at the bottom of the slide, I'm just showing you stills from that movie. So here is the first frame immediately after oocyte entry. I have a webby isotropic network, much like the network that I saw in the young animals that had never ovulated. However, after a while, I now see those beautiful parallel bundles that I have been talking about. Okay. And so the nice thing about Fibril tool and being able to visualize this in vivo is we're really able to tease out when is this organization occurring. And so by measuring cell length as a metric for cell contraction, so as the cell contracts, it shrinks in length, I'm able to determine when is contraction occurring. And using Fibril tool, I'm able to say when is the increase in actin organization occurring. And what you can see is as there is an onset in contraction, so as the cell length decreases, that's when I start to have an increase in actin organization. And shown another way, if I look at the slope of the line for the increase in organization, you can see that really peaks right as the cell begins to contract. So now we know that this organization requires ovulation. So the oocyte has to enter and stretch out the Spermatheca, but it really coincides with contraction. So it isn't just that cell stretch alone is enough to take this isotropic network and stretch it out in alignment. It's something about the onset of contraction appears to be important. We're also able to use this tool called Orientation J to look at how the actin cytoskeleton becomes oriented within each cell. So Orientation J is used to color fibers according to their orientation. And what you can see here are warm colors indicate about 90 degrees and cold colors are about 0 degrees, so horizontal. And not only are parallel fibers produced, but they are always oriented along the long cell axis, such that cells which are horizontal, such as this one you can see right here in the front, have a blue color, whereas other cells which are oriented vertically have a red color. And again, this occurs the same way in every animal always. Okay, So now I think we have the answer to our first question, which is when during ovulation does this organization occur? And it really appears to coincide with contraction. So then our next question is, what drives the production of these bundles and what controls their alignment? And so sort of our first hint at what might be happening here, in addition to the timing, is that we observed that there was a difference in actin organization between minimally stretched and maximally stretched Spermatheca. So sometimes during the first ovulation, instead of the entire oocyte getting in there, only a small fraction gets in. I'm showing you an example of that at the top here. So you can see the Spermatheca appears smaller because a smaller piece of the oocyte entered. And in that case, the actin network undergoes some changes, but does not become as organized as the Spermatheca here, which appears larger because a larger oocyte stretched it out. And you can see this graph here across multiple animals, is that there's a correlation between the Spermatheca, which were stretched more, and those that became more organized. And so our hypothesis for this is that something about cell stretch is actually increasing contractility. So cell stretch itself isn't responsible for aligning the network, but is perhaps modulating the amount of active myosin and therefore the amount of contraction. One explanation for this is that there is a mechanoperceptive rho gap, SPV1, which has been identified by the Lab, one of our collaborators. This rho gap is able to perceive membrane curvature, shown here, such that when the Spermatheca is unoccupied and in that really shrunken conformation, the membrane is very curvy, and so this gap stays there. But then upon ovulation, when the oocyte enters and the Spermatheca becomes very stretched out, the rho gap is removed from the membrane and you have increased contraction. So the takeaway from this is we know there's at least one mechanism where cell stretch can lead to increased contraction. So another way to more directly impact contraction, to try to uncouple cell stretch from actual contraction, is through taking advantage of what we know PLC1 does. So PLC1 is the phospholipase C responsible for calcium production, which increases myosin light chain kinase activity, leading to increased myosin activation. So if we get rid of PLC1, we don't have any calcium transients produced. Additionally, we shouldn't have any cell contraction. And so now I'm going to show you an ovulation video of a PLC1 null animal. This is much like the video that I showed you earlier, except now there won't be any calcium produced. And so now we get to see what happens to actin organization in the absence of calcium. So now, although we had entry, don't really have production of those parallel fibers. It sort of just sits there and wiggles around, but does not become organized like we saw earlier. Okay, so our hypothesis is then that PLC1 produces calcium, that calcium then, through its interaction with calmodulin, activates myosin light chain kinase. This increases the amount of phosphomyosin, and that something about active myosin is important for producing these organized bundles. However, to be honest, this is really a very simplified view of all of the things that calcium does within the cell. So it's possible that in addition to influencing the amount of active myosin, calcium could have some other important interactors. And so to really test, okay, is this something specific to the amount of active myosin, or is this something else that calcium is activating? We turned again to our knowledge of how Spermatheca contractility is regulated and looked at what happens if we get rid of the myosin phosphatase. So now we would hypothesize that in the absence of PLC1, we would have depressed activity of the calcium side of the pathway, right? So we don't have calcium production, so we should not have as much myosin light chain kinase activity. However, if you get rid of the off switch, the myosin phosphatase, you now would be able to unmask a PLC1 independent pool of active myosin and therefore increase the amount of active myosin present and hopefully, if we're correct, rescue production of actin bundles. And so we did just that experiment to see what would happen. Okay, so in the top here, I'm going to show you the wild type movie that I showed you before, where we saw actin organization coinciding with contraction. And on the bottom here, I'm going to show you the PLC1 null animal that I showed you before. This animal does not produce calcium transients and will not contract. So the actin network will stay very isotropic and disorganized. And here in the center, what we've done is in the PLC1 null, the animal that lacks calcium signaling, we've also knocked down the myosin phosphatase. So now any amount of active myosin that's present will be able to stay active, and we should increase the overall amount of active myosin and potentially be able to rescue parallel bundle production. So let's watch and see what happens. And what you can see, sure enough, this animal definitely has more active myosin than the animal at the bottom, and definitely has rescue of parallel actin bundles. And quantified here on this graph, you can see wild type at the top is the most organized, whereas without calcium signaling at the bottom shown in green, there is really no organization occurring. And then I do get a slight rescue if I knock down the myosin phosphatase. So I've increased the amount of active myosin, and I do see rescue of my actin network organization. Okay, so now we have the answer to our first question. This dramatic change in actin organization really coincides with contraction, and unsurprisingly, given when it happens, this requires myosin activity. So myosin really appears to be driving actin bundle production. However, we did make another observation based on this data here, and that is that while we do have an increase in bundle production, as you can see, the rescue isn't perfect. So this orange line here is nowhere near as organized as the wild type line here shown in purple. And yes, it could be true that we don't have the same amount of active myosin. However, we are also not necessarily recapitulating the wild type spatial temporal activation of myosin. So it might be important not just how much active myosin is present, but exactly when during stretch and contraction do you have peak levels of myosin activity. So looking at this further, we noticed that in addition to not being as organized, what we really noticed is that there was a failure to produce the alignment along the long cell axis. So again, I'm showing you data using orientation J, and in the top here, all of my fibers from a single cell are oriented in the same direction. So they're all the same color, oriented about 90 degrees vertical, all red colors. However, in the same cell, but in a different animal, this animal now is the PLC1 null that has been treated with MEL11 RNAi to increase the amount of phosphomyosin, I can see, yes, in fact, there are very clear bundles produced, but they're no longer perfectly oriented along the long cell axis. And I have populations of differently oriented bundles. All right, so myosin activity is required for bundle production, but something about the proper activation of myosin is important for producing the stereotyped organized network. All right, back to the slide. So in order to more directly probe what the role of myosin activity is on actin network alignment, we used a few different techniques. So instead of disrupting calcium, we really wanted to hammer how much active myosin that there is. So we did this in two ways. One, we either got rid of the myosin light chain kinase, so that should really depress myosin activity and keep myosin in this inactive conformation, can't interact with actin, and can't produce contractions. We also got rid of the myosin heavy chain itself, so no myosin present at all. And then the other thing we did to really ramp up myosin activity to see what happens if we go in the other direction and have highly elevated myosin activity. What happens to actin organization and bundle alignment? And so to accomplish that, we got rid of the off switch, or myosin phosphatase. In the absence of myosin phosphatase, any myosin that becomes phosphorylated and active stays that way. And so you just increase and increase and increase and constitutively contract. Okay, so let's see what that looks like. What I'm showing you here in red and in green are results from phalloidin staining, so that's cut and fixed animals, and then our in vivo marker is in green. And I'm just showing you both of these to demonstrate that everything we've observed, we can observe in multiple ways, and it isn't just that our actin labels are influencing the experiment outcome. So on the top here are control animals, and as I've been saying all along, you can see that there are parallel actin bundles present, and they're oriented along the long cell axis in each case. And then if I get rid of myosin entirely, I have this really flaccid Spermatheca. You can see the valve shown here in both cases really stays open and flaccid. It's not able to close at all. And if you look carefully at the actin structures, they remain very tortuous and webby and resemble the actin organization in the young animals that had never undergone an ovulation, even though these are old animals and the Spermatheca has become stretched out. So really, in the absence of myosin, we don't get any organization of the actin network, really demonstrating that myosin is important. Okay, and so then here's kind of the fun part. Now, if we get rid of the off switch, the myosin phosphatase, and really constitutively increase contraction, we get this crazy phenotype where we now have really huge, prominent actin-myosin bundles. And what's interesting to me is that in addition to production of really large bundles, you can see that these bundles get pulled into different orientations. So there's a nice example there. So this sort of gave us insight into how it might be that myosin activity is important for regulating the alignment along the long cell axis. All right, so throughout this talk, I've been talking a lot about actin and myosin, but primarily just showing you what actin looks like. And we're really interested in what does myosin look like in this network as well? How is myosin organized in the Spermatheca? And so to answer that question, we looked at both actin and myosin in animals. So here I'm showing you actin in red and myosin in green. And again, at the top is a control animal. And what you can see are those beautiful parallel fibers running along the long axis of each cell that you've seen the whole time. And then if we use a line scan and look at fluorescence intensity, what you can see is everywhere there's a peak in actin, indicating an actin fiber, there's also a peak in myosin. So these are really tightly associated into these actin-myosin bundles. Additionally, you can see that there aren't really any differences in myosin accumulation along the fibers. In other words, myosin is homogeneously distributed throughout actin-myosin bundles. It doesn't seem to accumulate anywhere. All right, so what happens if we get rid of the myosin light chain kinase? So now we've really depressed myosin activity. Well, unsurprisingly, myosin now remains largely cytosolic. So you can see that through a line scan. You can see that now where there's a peak in actin, there's no longer a peak in myosin. So this makes sense to us because without phosphorylation, myosin stays in that closed off conformation. It's not able to form filaments and interact with actin. So now it just stays in the cytosol, floating around, and isn't helpful. So the next thing we wanted to look at is what happens if we go the other way and we really increase the amount of actin myosin. Well, I had a hypothesis, and what I had imagined is that we would get a case that looks much like this top graph here, except perhaps we would have larger peaks in green, indicating hyper recruitment of myosin into these actin-myosin bundles. Much to our surprise, however, we got this, which is totally unexpected for us. So instead of having increased myosin recruitment to these bundles, homogeneous throughout the bundles, we now have these really light, really bright foci being formed. So if you look here, you can see that there is this band of myosin that forms and also appears to be connected across multiple parallel actin bundles. So this was very intriguing to us. We wanted to understand, how is this forming? How is it that myosin goes from presumably something that looks like this top animal here, where it's homogeneously distributed throughout, to then when you really increase myosin activity, forming these bright myosin foci that are also interestingly aligned across multiple actin bundles, suggesting potentially that myosin is interacting across bundles. And to answer that question, we turned to in vivo visualization of this. So this again is an ovulation movie, much like the ones I've shown you earlier. Now I'm showing you myosin in green, actin in red, and where actin and myosin co-localize, you'll see yellow. So the top is going to be a wild type movie, and the bottom is what's going to happen if I get rid of MEL11. MEL11 again is the myosin phosphatase or the off switch. So in the top movie, the oocyte is going to enter, stretch out the spermatheca. Spermatheca contractions are then going to culminate in exit of the then fertilized embryo into the uterus. So it's all going to happen quite fast. I've sped it up so that the bottom movie plays at a more reasonable rate. So what's going to happen at the bottom movie is that after oocyte entry, the valve is not able to open and the embryo becomes trapped. So because I've gotten rid of the off switch, the phosphatase, I now have constitutive contraction. So the tissue is going to continue to contract and contract and contract, and we'll be able to watch what happens to myosin organization throughout this process. So again, top movie, entry, culmination, contraction, and exit, and the spermatheca goes back to that closed conformation, and now crazy things are happening on the bottom. And I will explain. What? Okay, I didn't prepare you. All right, so we're gonna watch, I know that played quickly, we're gonna watch it again. On the top, we have entry, we have initiation of active myosin and contraction both through the Rho pathway and through the calcium pathway. this leads to myosin recruitment into actomyosin bundles, pulling on those bundles, and then exit because the tissue contracts. However, on the bottom, it's unable to exit the embryo. The reason it's unable to exit is because the valve stays really tightly closed. So as contractions increase and increase and increase, instead of the valve nicely opening, what actually happens is these basal actin bundles pull apart, and the tissue actually rips as the cell junctions break, and now the embryo is released into the body cavity of the animal, which is not where it's supposed to be. Okay, so now if we watch that bottom video again, you can see at the basal surface there, it really starts to pull back right about now. Yep, and now the whole thing is going to split open like that. Okay, so the movies play a little bit fast, and maybe it's easier to see what's going on if we look at individual still frames. So at the top here, I'm showing you what a wild type movie looks like, the same one I showed you on the previous slide. And what you can see is initially myosin is largely cytosolic, and you can just see some actin fibers, but it really just looks like a green haze. And the reason for that is because until contraction is initiated, myosin is in the cytosol. It's not pulling on the fibers. And then once contraction is initiated, it's sort of hard to see actin network organization because the tissue really rapidly contracts as the oocyte is pushed out, or the embryo, I should say, is pushed out into the uterus. However, on the bottom, now I've gotten rid of the myosin phosphatase, so now I have constitutive contraction. And what you can see here is initially it looks much like the top frame, where I have actin fibers and largely cytosolic myosin. However, even just 3 minutes and 20 seconds into the movie, if you look very carefully, you can start to see small myosin foci along the actin fiber. And these foci really increase in intensity over time. So here, just a few minutes later, you can see that they're much brighter and much more prominent. Additionally, what you can see is they start to align. So as you have multiple foci, as they come close to each other, they align together. So it looks like that myosin foci on parallel fibers are actually somehow able to interact with one another such that they become aligned across multiple bundles. We were also really fortunate in this movie able to catch another cool thing, whereas in this case here, those two actin fibers or actin bundles have just retracted backwards. And they're able to retract up until reaching, it's a lot of arrows, a band of myosin shown there. So it really looks like myosin, again, is interacting with other myosin foci on parallel actin bundles, such that these two fibers are able to retract but then stop once they reach this continuous band of myosin. And this process continues all the way up until this basal layer pulls all the way back as the fibers really increase in contractility. And then as you saw in the movie, this finally culminates in such intense contractions that the tissue rips apart and the embryo is just released into the body cavity of the animal. It is perhaps easier to see how these myosin foci are coming together if we look at a chymograph. So on the bottom panel here, I'm showing you chymographs from 4 different cells across 4 different ovulation movies. And let's just pick an example. So if you look right here, what you can see is that at the beginning of this movie, so time is along the bottom axis, the yellow is very homogeneous, meaning that there is both red and green present in this cell. And then as the movie plays, you can see right where I put the arrow, I have these 2 myosin bands that then sort of come together. And so that is why the myosin foci appear to be becoming brighter and aligned as we play this movie throughout time. Okay, so in conclusion, what I've shown you today is that calcium signaling is tightly regulated during ovulation. So we only see those peak in calcium transients produced really at the onset of tissue contraction. Additionally, what I've shown you is that actin network organization coincides with contraction. So cell stretch itself, while required to initiate contraction, is insufficient to align the network. So it isn't just that I've taken this isotropic network and then pulled on it and that is what's causing the alignment. Instead, it's a very active process and requires myosin. And then finally, what I showed you through multiple ways, that myosin activity, it really appears to be finely tuned, such that if you have a smaller fraction of oocyte that enters the spermatheca, you produce less contraction, you have a slightly less organized network. If you go the other way and you really ramp up the amount of active myosin, you then have really thick bundles that are no longer oriented in the proper direction. We think that's because myosin is able to interact across multiple actin bundles and really pull them into different directions inappropriately. And what I'm showing you here on the bottom is just that, again, if you have either depressed or really increased myosin activity, you not only alter actin organization, but you alter actomyosin network organization, and myosin is now differentially recruited to and organized into these bundles. Okay. And so, future work. What are we interested in doing next? Well, I focused so much today on actin and myosin, but really there are a lot of other proteins interacting with actin. So actin participates in more protein-protein interactions than any other protein in cells, and we know that it's not acting in isolation. So we're now interested in identifying other actin interacting proteins that are influencing things like the orientation of these actin bundles and other actin interacting proteins that are affecting their connectivity. So these will also be important for determining the contractile properties of this tissue. And one of the approaches we're taking to do this is an RNAi screen. So another really great thing about working with this animal is because they're transparent, they're really easy to observe. So what I'm showing you here is the Spermatheca expressing GFP. And so at the top here is a control animal, and there are two Spermatheca lit up with GFP. One Spermatheca appears very round. That's because this Spermatheca is occupied by an oocyte, whereas this other Spermatheca here appears very small and compressed, and that's because this Spermatheca is unoccupied and does not have an oocyte present. So in wild-type animals, these animals are constantly ovulating. Each animal produces 300 offspring, and so over time, the Spermatheca becomes occupied and then contracts and becomes occupied and contracts. So if we take a snapshot and look at a whole population of animals, we see that a small fraction has an oocyte or an embryo in the Spermatheca, so it has that rounded occupied conformation, whereas most of them are unoccupied because they're constantly contracting. But we reasoned if we disrupt a gene that's required for contraction, we should then see a shift, and now we should have a population that has most of the oocytes or embryos trapped in the Spermatheca, or if they're not trapped, at least spending a longer time there because tissue contraction is not initiated at the right point in time. So in this example, I've shown you what happens if we get rid of PLC1. PLC1 is the phospholipase C responsible for producing those calcium transients that then initiate contraction. So we knew that that gene would definitely disrupt contraction, and that's what we use as a control. And if you look at the wild type side, there are many worms, and each animal has, most of them have unoccupied Spermatheca, and then if you get rid of PLC1, we now have a population where nearly every Spermatheca, or 100% in this image, are occupied, so it's not able to contract. So we're now using this to screen actin interacting genes to identify ones that shift the population so that more of the animals have an occupied Spermatheca, so more animals have a defect in contraction. All right. And with that, I would really like to thank everybody who made this work possible, especially the Cram Lab for making it a really wonderful place to work. And I would like to make a special shout out to my advisor, Dr. Aaron J. Cram, who's been an excellent advisor, and also to past graduate student Ismail Kovacevic, who made the initial observation that the actin organization in young animals is different from the actin organization in adults. Additionally, this work would not be possible without our collaborators at the Zydelbar Lab, who have just been great for bouncing ideas off of and providing worm strains. Additionally, our funding and other providers of worm strains from the CGC and the Sherwood Lab. And with that, I would like to take any questions.
Thank you, Dr. Alison Wirshing, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window. Type your questions into the box that appears on your screen, and click on the send button. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, what is the role of cell stretch in actin organization?
That's a great question, because I know in other systems it's been shown that if you increase cell stretch, you increase the production of actin bundles. And in this case, we do think that cell stretch is important, but not sufficient. So cell stretch is really important for triggering contraction. And we also have some data to suggest that the degree of cell stretch correlates with the degree of actin organization. but we think that's because cell stretch is then increasing the amount of active myosin. It isn't just that as you stretch this network more, it becomes more organized.
Thank you, Dr. Wirshing. The next question is there a way to increase or decrease cell stretch?
Yeah, this is a question we get asked all of the time, especially by people who work with really interesting in vitro systems where they can grow cells in different surfaces that allow them to do things like compress the cells and stretch the cells and poke them with things. One of the nice things about having an in vivo system is that we know that everything that's happening happens for real in an unperturbed system. But one of the downsides is it's much more difficult to manipulate. So it's hard for us to get in there and to do things like stretch the Spermatheca in the absence of oocyte entry to clearly demonstrate that it's not something special that oocyte is doing and more a mechanical reaction to just being stretched by this object and then producing contraction. One of the things we have been able to do is to knock down different genes that influence the size of the oocyte, so that regulate oocyte streaming and produce oocytes that are twice the size, or smaller oocytes. And in that case, we do see that there is a change in actin organization. It's somewhat subtle, and it's very much like the data that I showed you where when a smaller piece of the oocyte gets in there, it does not become as organized. It's more difficult for us to manage to get a really large oocyte in there and stretch the Spermatheca out more, just because those oocytes have a hard time entering and those events are not very frequent.
Thank you for that answer. The next question is, what are the actin bundles attached to?
So that's something we're actively pursuing. Typically, you see actin stress fibers ending in focal adhesions and interacting with adherence junctions. So we know that focal adhesion components are important in the Spermatheca, but they do not produce those really stereotypical, really prominent focal adhesions that you see in adherent or migratory cells. Additionally, we know that adherence junction components are important for actin network organization and maintenance of the actin network over time. So if you disrupt adherence junctions, you get disorganization of the actin network, and this also appears to get worse over time, suggesting that they're important for actin organization and perhaps anchorage itself. Interestingly, however, the adherence junction components are apical, and these actin bundles that we're looking at are basal, and they don't appear to come down and touch the apical surface. They don't appear to be directly interacting, and so we have done this RNAi screen and have a few candidates we're interested in looking at to really find out what is it that's connecting these fibers to something, potentially at the apical side.
Okay, it looks like we have time for one more question. What are your next steps?
Yeah, so as I alluded to at the end of the slide, we've done this big RNAi screen to identify actin interacting proteins that are important for organizing this network. So the thing that I'm really interested in, again, is something that would potentially influence actin network connectivity or just connection at the junction. So another actin interacting protein that helps to potentially cross-link or bundle actin filaments. We've identified filament in a previous screen, and if you're interested, Dr. Kovacevic has a lot of work published on that. So that at least is one actin interacting gene that plays an important role, but we're looking to identify others.
I would like to once again thank Dr. Alison Wirshing for her presentation. I would also like to thank Labroots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through December 2017. You will receive an email from Labroots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.